So let's move on to keep to the time schedule. I'm happy now to hand over to Tien from the National Central University in Taiwan, and he will tell us about crosstalk between VHL mutation in kidney cells and in the tumor microenvironment. So, Tien, I give the word to you.
Okay. All right. I couldn't
just share your screen first.
The screen. Yeah.
Can you click on the share screen icon? Just beside
the... Here it is.
Yeah. Now we see that.
Yeah. Okay. Thanks for the organizing committees, and greetings to everyone from Taiwan, which is 30 past midnight. I just had 2 coffees, so I should be fine. So, so I was trained as a developmental biologist in Osaka. So I like to look at things in the tissue context. So, so I'll tell you a little bit about our recent work on the microenvironment, and we begin to dissect this very complex area. And I'll tell you about this briefly. So, number of years ago, we generated VHL, conditional knockout, using HOXP7 pre-GFP. Andy McMahon, when he used this, when he generated this HOXP7 expression pattern, it was shown in collecting dots. But in our hand, we found in the adult, actually HOXP7 is expressed in the cortex. For example, here, using the wild type, just the GFP without VHL, it's expressed, you can see the expression is in cortex, even in the proximal tubules, and also in the medulla. This looks like Henry's loop. But using GFP, we can trace the mutant cells. For example, in the cortex, you start seeing the piling up of the epithelium, it looks like an adenoma. And you also start seeing the appearance of clear cells, also this very clear adenoma. And in the meantime, you also start seeing surrounding these abnormal tubule cells, there are infiltration of immune cells. So at the tissue level, what you see is the clear cell clusters of these abnormal tubules, and also the fibrosis, and a lot of immune cell infiltration in this particular area. We call it an inflammatory focus. And also here's another example. There are also a lot of vasculature, as you expect. So we think this is actually a very good model because we're seeing very severe inflammation and fibrosis in association with some hyperplasia and cyst formation. So we think this is a very good model for studying the cancer-associated inflammation. And then a couple of years back, we showed that the VHL knockout or inactivation in the kidney tubule cells actually causes ER stress. And that's because of the metabolic imbalance. And with this ER stress, there's overexpression of or activation of JNK. So you can see the increase of phospho-JNK. And this is further increased if you make this ER stress worse by tunicamycin. So then we stain the tissue. So we want to verify this. So this is mouse tissue, wild type. There's no phospho-JNK. In the knockout mice, there is JNK as expected, phospho-JNK in the abnormal tubule. But surprisingly, we also observed very prominent expression of phospho-JNK in the nearby vasculature, and even in the more distal area. So the vasculature is, so to speak, inflamed. And this is also can be observed in the human CCRCC samples. This is adjacent normal tissue with some inflammation. So you can see the phospho-GNK expression in these abnormal tubules. This is almost like nascent clear cells. But in the cancer tissue, CCRCC, you see the phospho-GNK expression exclusively in these blood vessels. So this is peculiar because the knockout happens in the epithelial cells, not in this endothelium. So there must be some kind of secreted factors produced by the VHL mutant cells that's influencing these endothelial cells. So then we isolated these endothelial cells, and immediately after isolation, we put them isolated mRNA and without any culturing, subculturing. So we immediately put them to RNA-seq. So what turns out gene expression pattern were quite different. And again, these endothelial cells does not have any known genetic manipulation. So these overexpressed cells can be grouped into different functions, and most of these actually are involved in immune response. And you can also see the production of IL-6 and cytokine secretion, but you also see these are endothelial cells. Cell adhesion changes, and cell shape change, and cell migration. If you look at individual genes, you can find genes that are involved in immune cell capture and recruitment, rolling, and for example, selectin E, selectin T, ICAM1, ECAM1. Involved in inflammation, for example, kidney injury molecule 1, leukocaine 2, IL-6, IL-33. And very interestingly, there's also a group of genes involved in endo-MT. For example, SNAIL1, ZEB1, these are transcription factors associated with mesenchymal transition, collagen 1, and bimentin 1. To verify this in the mouse tissue, if we stain for ICAM1 in the wild type, there's nothing. In the mutant, you can see very high expression in these abnormal vasculature here. And this also happens in the CCRCC samples. In the adjacent normal, there's nothing. And in the tumors, there's expression only in the vasculature. SNAIL1. SNAIL1 is a mesenchymal marker. It's a transcription factor. In the wild type, there's no expression. In the mutant, it's expressed in the vasculature surrounding the tubule. And the vasculature localization is confirmed if we use CD31, which is mostly in the blood vessels. In SNAIL1 double staining, you can see the SNAIL1 is expressed in these vasculature. So, endo-MT, in fact, it's a very important cellular mechanism for endothelial cell activation and tissue fibrosis. So as I mentioned, we suspect this the changes in the endothelial cells actually came from secreted factor from the VHL mutant epithelial cells. So using the IPA upstream regulator analysis, we found actually oncostatin A signaling is the most prominent, most significantly upregulated. And there are other pathways. In fact, some of these are actually the same as oncostatin M, like STAT3. STAT3 is activated by oncostatin M. And IL-6 is a very very much related, and we'll come back to that, and oncostatin M receptor itself. So it's a very prominent signaling pathway in this phenomenon. And we verified the oncostatin M expression together with GFP-marked mutant cells. And oncostatin N is an IL-6 family of cytokine. It's a very potent pro-fibrotic and tumorigenic cytokine. And in the human settings, actually, high expression of OSM and OSMR is associated with decreased survival. So to make the long story short, when we combine the conditional VHL knockout with the oncostatin M receptor knockout, all the indices are reduced. For example, this secreted selectin E is activation marker of endothelial cells. It's reduced from the VHL conditional knockout. CD31 positive cell number is reduced, and the F480 staining, that's stained for the macrophages. So the macrophage infiltration is also reduced. And these inflammation foci number is also reduced. And importantly, this dramatic vasculature, abnormal vasculature in the VHL mutant is rescued back to the wild type shape. And the vascular leakage shown by the injection of FITC labeled dextran in the mutants, VHL mutants, very much diffused out, but double knockout rescue their phenotypes. And importantly, proliferation index, Ki67, is reduced, both in the immune cell population and in the tubule population. So actually, oncostatin N signaling is also involved in the hyperplastic phenotypes. And as I mentioned before, this IL-6 itself is actually induced by oncostatin N, as shown here. IL-6 is a very potent pro-inflammatory cytokine, and itself can induce macrophage activation and tumor progression. So if we inject anti-IL-6 antibody into the mouse, we can also reduce these inflammatory foci, and the injection itself has no effect, no adverse effect on wild type. And also here we showed after blocking IL-6 in vivo, the total macrophage number is rescued. Interestingly, it doesn't change the composition of M1, so-called M1 macrophages. M1 is an anti-tumor pro-inflammatory macrophage, but in the knockout, VHL knockout, the M2 as well M1 was increased, but double knockout, when we inhibit IL-6, actually the M2 macrophage will be reduced. Now, M2 is a pro-tumorigenic and anti-inflammatory macrophage. So this signaling, actually, OSM-induced IL-6 can change the macrophage composition. And what does this M2 macrophage do? So this is a simple experiment when we have using HK2, which is a non-cancerous tumor kidney tubule cells. We knocked out VHL. There's the morphology didn't change, but if we knock down VHL and co-culture with macrophages, remember these macrophages can be induced into the M2, and these tubule cells actually start changing, and there's an EMT type of transition going on, and this can be shown by the reduced eckaterion and increased enkaterion, and so forth. So to summarize, what we show is that when there's a VHL mutation, these newly formed hyperplastic cells will secrete oncostatin M. Oncostatin M can induce neuroangiogenesis, and then they can activate endothelial cells. These endothelial cells activated can capture the immune cells. They can promote the extravasation of the macrophages, And they also secrete IL-6. IL-6 can come back and further induce the endothelial cells. They can also induce the polarization for M2 of the macrophages. And these induced activated M2 macrophages can come back and induce the further tumorigenesis. What I didn't show is this actually through the secretion of CCL18 and TGF-beta. So I think this actually provides, I think, for future, maybe a way for preventing or treating early stage CCRCC in association with the tumor-related inflammation. So with that, I'll stop here. And these work are actually done by two very capable graduate students, Young and Hui, and also very capable postdoc. So I'll stop here. Thank you very much.
Thank you very much. It's a very in-depth and very interesting characterization of the crosstalk and affecting the microenvironment. I think I take up your last point. I would argue that not only the early stage CCRCC, I think this network is interesting, but potentially to therapeutically manipulate the microenvironment in the context of immune checkpoint therapy towards maybe in some patients towards a more favorable environment that are more conducive to checkpoint then.
Right. Yeah. Thanks, Ian. So yeah, that's a very good point. What I didn't show is actually that microenvironment generated by this VHL knockout, when we injected a melanoma cell line, this actually promotes the metastasis.
Okay, interesting. Yeah. And one last question, I think, before we move on, is from Rachel Giles again, who asks whether you think that the endothelial cells are maybe de-differentiating towards a hemangioma-like phenotype. And would IL-6 inhibition be something to think about therapeutically in that context? Or do you think this is maybe an organ-specific crosstalk that you've identified?
We didn't look specifically, but this changes, surprisingly, these changes in the endothelium, I think, is systemic. So we look at the skin, the blood vessels in the skin, they also change. So that must be the secondary effect. We think that might be IL-6 secreted by the endothelial cells that spreads throughout the system. So if that will be part of the hemangioblastoma, we don't know. But that's a very interesting question.
Actually, I just literally saw that one more question just came in, and that is that asking about the reason for the HOX B7 driver. Why did you choose that? And when there are other drivers as well, of course, that are maybe more proximal tubule or cortex specific.
You probably know this more than I do. So when we started doing this, there are a lot of knockouts already done. So we just wanted to choose a different cell populations, and turns out it hits. We probably hit some special cell types. So we're trying to figure it out.
Okay, very interesting. Thank you very much.